So this unit is going to be a lot of handwritten notes, but I'm also going to do a few lessons in a way I normally don't teach. But I'm going to do some of the sort of just PowerPoint slide and present, because you'll see a lot of that in university biology particularly. You don't see it so much in chemistry and physics, where it tends to be somebody just writing on the board. I think that the PowerPoint's kind of the most dangerous of, because it's so tempting and so easy just to be like, I can just download the notes, I'm just going to watch along. So I'll talk about some strategies for that. But today we're just going to do a handwritten note together. All right, so I know it's already written above, but I want to write it. Okay, in case you don't recall, DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. Ribo refers to a ribose, which is a type of sugar. Obviously, the fact that this says acid tells us that what functional group should be present? Yeah, so we're going to have a carboxyl group present, right? Right? It's actually a bit of a variation, but that's okay. But there it is. So, no. Oh, come on. What's Italian for sugar? It's so good. Do you know? Zucchero. Isn't that a great word? Zucchero. Not zucchini. That's a different thing. But zucchero. Anyway. So DNA is a polymer. And what's another word we use for polymers sometimes? Yeah, it's a macromolecule. Made up of monomers called, what's the monomer of a DNA molecule again? Nucleotides, good. All right, and nucleotides themselves have 3 parts. You want to remind me what the 3 components of a nucleotide are? Yep. A phosphate group, very good. Next. A nitrogenous base, very good. And thirdly? Very good. Specifically? No. It's right in the name. Specifically, deoxyribose, which is ribose missing an deoxy. It's missing an O. Yeah, an OH as it happens. So the sugar is deoxyribose. Good. Mr. Matola, how are we doing, my friend?
Sorry about
No, don't be. What can I do for you?
Able to perform emergency surgery in a volleyball match?
Probably. For when?
Tomorrow.
Yeah, you bet. I'll do it right after school. Can you bring it down here so I can't forget?
Yep.
Yeah, you bet. All right. So what's that actually look like? Well, what do sugars look like? We know that. A ribose is a pentose, which means it's a 1, 2, 3, 4, 5 carbon sugar. And it's going to form a ring like this. So let's actually draw this. The ribose itself, the sugar group, it's going to look like this. And we're going to number the carbons. All right? C1, C2, C3, C4, which is going to connect back to the oxygen. Oh, that looks terrible. I'm going to try again. Okay. Again, if I've never told you this, I get very nervous when I have to draw. Makes me very uncomfortable. I'll do it symmetrically as I go. Okay, there we go. And then up here would be C5. And that should look familiar, having one carbon sit above the ring, right? We've seen that with sugars before. With our hexoses, glucose has one carbon that sits above the ring. It's a ketose. And then you remember we had our aldoses like fructose, which actually has two carbons that sit above the ring, right? So this has one carbon that sits above the ring like that, carbon number 5 specifically. And from carbon number 2, we have a hydrogen present. Carbon number 3, there's an OH group down here. There's hydrogens coming off the others as well. I'm going to highlight these only because this has the OH group, and then this is called deoxyribose, because normally there would be an OH group here on ribose, but this one is missing one. Does that make sense? You don't have to have that. Well, I guess you should know that. That's something you should know. And then we know that this is connected to a nitrogenous base, which is always connected to carbon number 1. And there are 4 different nitrogenous bases. What are they? Guanine, thymine, and adenine. Good. So guanine, cytosine. I don't think I've ever written these in this order before. Adenine and thymine. Yeah. I usually put adenine and guanine together, and then cytosine and thymine together, and we'll talk about why in a minute. That's okay. And then what's the third component? Correct. Okay. And that's going to connect off of carbon number 5. And what's a phosphate look like? It's phosphorus surrounded by oxygens. Very good. So we've got the oxygen here. It's making a single bond to phosphorus, which is making a double bond to another oxygen, and single bonds to 2 others. And this one happens to be an OH group. Guess we'll make note of that.
I didn't work for this, Mr. Orlando. Like,
You were unaware we were building higher.
Sorry. You'll be all right.
no. It's not gonna look pretty now.
If you don't have space, just use the lasso tool, select it, and move it.
This is true. You could do that. I'm just going to...
Are there any other OH groups coming off of, like, carbon 4 and 3?
Do we... I don't love that. But that's okay. We're gonna stick with it.
Where's the stuff happening? Sorry.
So we'll make a note. We'll say that the lack of an OH on carbon 2 makes this deoxyribose. See, not CO2, just carbon 2.
Is there supposed to be an OH group on carbon 4?
No. There is an H down there. Again, there would be an H here, there'd be an H there. There'd actually be another H on this side and another H on that side. I'm not drawing all the H's. I'm only highlighting the H on carbon 2 because what is conspicuously not there? OH. Does that make sense? So obviously fill in everything with hydrogens as you normally would, but we just want to make note of the fact that carbon 2 lacks an OH group. Did that answer your question? Mm-hmm. Good. This is only worth mentioning because later on we're going to talk about the structure of a very similar related molecule, which is what? RNA. And it's ribonucleic acid. And in lieu of deoxyribose, it's built around ribose, just regular old ribose. And it actually, that one small difference makes an enormous difference, has very different chemical properties. Not very, well, yeah, no, very. I stand by the very.
A double helix, it's a single squiggle.
So if you want to say that, you can. You don't have to. So with that OH, it would be ribose, the foundation of RNA. If you recall the bases, we'll talk about it now. If you recall the bases, they're divided into two categories. You have the purines, and you have, does anyone remember the other ones? Remember this? Purines and pyrimidines. Very good.
These bases are for RNA?
These are for DNA.
But thymine, adenine, cytosine.
No, but they're
Yep.
grouped into different
The
groups.
purines are adenine and guanine.
Oh, right. I remember this. It's great. This is great.
If you're trying to remember which are which, I always remember what we came up with, or I don't know if my teacher told me this or we came up with it, but what we always remembered it by when I took biology a long time ago was pure as gold. Pure as gold. Pure, purines, as gold, adenine and guanine. And then the other ones are cytosine, uracil, don't worry about it yet, and thymine.
I had a whole argument with Mr. Trush about uracil.
Fascinating. What was the argument about?
Because we were doing the project we had a bit ago where we had to create the molecules.
Okay.
And I was talking about these, and he's like, "Oh, you're forgetting about uracil." And I was like, "Yeah, but we're not doing RNA, we're doing DNA." And he's like, "It still counts." And it was a whole thing.
Did you have an arm wrestle?
No, he beat me.
He would probably beat you. Yeah. Structurally, what makes purines and pyrimidines different? We'll look at the full structures later, but the main thing you can know for now is purines form a double ring. They kind of look, that's the worst double ring anyone's ever seen, but that's okay.
How am I supposed to remember this one? You just said that.
I will tell you I remember. Pyrimidines make a single ring, and I've never seen a double-based pyramid, have you?
No.
Ah, see? See? In 26 years, I still remember it that way.
Do they always make pentagon?
We'll look at the actual structures later. These are very crude drawings, but that's okay. Okay, but now if we can pause. Don't worry, I'll come back to this. If you don't have it drawn, don't freak out. How do you make a polymer out of this? Where does the actual bonding happen? Okay. I gotta be honest, I can't interpret that one.
The OH at the bottom?
Okay, and what carbon's that on?
3?
Okay, and where else?
On the phosphate one?
Sorry?
On the phosphate one?
Yeah, and why are we looking at OHs?
Because each can go in oxygen.
What kind of reaction can happen there? Sorry?
The watery ones.
Yeah, not hydrolysis. Hydrolysis is the opposite. That's when you use a water molecule to break something apart, but the opposite, which is? Condensation. Good. So DNA forms a chain, or a polymer. So DNA polymerizes rather. Polymerizes? I don't know how you'd pronounce that. Anyway, polymerizes, i.e. forms a chain. Why indeed? when a condensation reaction So there's a long sentence, which we can probably just draw it and have it be better. But do we see that? I'll come back to this, so don't worry. But if a second nucleotide shows up, that second nucleotide can react this OH group to that OH group, and they can form water. Probably we should draw this, yeah? But let's draw it a little bit more schematically. Well, is everyone comfortable with that? Just to make this kind of... Is everyone kind of comfortable with that level of schematics? It's an OH group attached to a phosphate, and this is an OH group. And if another one comes along, we can have the phosphate group here, right?
Can you just scroll up like a tiny bit?
I would love to.
And show me places
I resent, on my own account, how messy I am. And what we're going to see, of course, is that the reaction actually doesn't happen within a single molecule. It happens here between these two OH groups, right? And what do we end up with? Well, we release water, right? And what else do we get? Well, now these are stuck to each other, right? So what will be between them? An oxygen. Very good. What kind of bond is that? An ether bond. Good. By the time we're done, we actually call it a phosphodiester bond. We can call it diester because this actually has a double bond oxygen. So we refer to this as a variation on the ether bond, where because this has a double bond oxygen as well, which I'm not bothering to draw, we call this a phosphodiester bond. Phosphodiester bond. We'll make a note there.
Wait, what has double bonds oxygen?
The phosphate group.
It's a double bond?
Yeah, if you go look at phosphate, right? It still has this.
Yeah.
So that would be considered not an ether bond then, but an ester bond,
Yeah.
right? And because it's also got this here, we call it a phosphodiester bond. And what was your question, Toby? Or
Where
comment?
did the base go on the bottom one?
Sorry, Toby, what's your question? Okay, try not to distract people.
Yeah, it's hard. I'm trying to rank here.
I always do this when I do drawings. I always just leave a massive gap to the left, and then it ends up expanding to the right in ways that I don't intend. It's the end of my story. Okay, now, we refer to the location on the sugar using this notation, prime notation. So for example, a bond, we would say like 5' and 3' instead of saying like on carbon 1 and on carbon 5 and on carbon 3. Does that make sense? So we say that this bond is between the 5' and the 3' of the two molecules, right? So if you look here, this sugar here is the 3', and or this carbon here rather, and this carbon here is the 5'. Do we see that? So it's a 5' to 3' phosphodiester bond. So on each sugar, on 1', i.e. on the carbon number 1, we have the base. On 3, we have the bond to the next nucleotide. And on 5', we have the phosphate, and that is bound to the previous nucleotide's 3' end. Does that make sense? This is what our sugar ends up looking like. On each sugar, we have the 1' contains the nitrogenous base. The 3' is making the bond to the next nucleotide's phosphate. We could even say that. So the 3' of one nucleotide is stuck to the 5' of the next, and the 3' of that one is stuck to the 5' of the next, and the 3' of that one is stuck to the 5' of the next, and so on, and so forth, and so on, and so forth. Right? So a single strand of DNA is just a chain of nucleotides. All right, ready for some great drawing? Yeah, you are. Okay. So what we're going to do is we're going to draw it this way. Okay. Which way do I want to start? Well, we'll start with 5' up, I guess. So we'll start with the very first. I'm going to draw circles to show the phosphates, right? And then we would have the sugar, right? And then maybe this one is connected to, I don't know, an adenine. And then let's label all these quickly. We won't label each one, but this would be 1', right? This would be 3', and this would be this one up here would be 5', right? Which is what's actually connected to. Okay. Then what happens? Well, there'll be an O between them, right? And I'm not even going to bother showing the O between them. I'm just going to, for simplicity's sake, show the next phosphate, because we know phosphate includes oxygens, right? And then it will go down to another nucleotide like this. And maybe this one's stuck to, I don't know, guanine. And on this one, again, that's the 5', that's the 3', that's the 1', right? What would we see next? Well, we'd go down, we'd have a phosphate group. There we go. And maybe this one's connected to another guanine. And then, I don't know, we go down like that, down like that. I'm just going to do, obviously you could do this for days, because how long does this actually go on for? Millions and millions and millions of nucleotides, right? What do we want these to be? Give me a code. What's next? T. Okay. Thymine. What after that? C. Okay. Cytosine. What after that? C again. Okay. There we go. And on the very last one of these, what would have to be down here? Well, an OH group, right? And on the very last one, what carbon is this? 3 prime. So at the 3 prime end, there would be an open hydroxide, right? And what would be present up at the 5 prime end? This very last one. Yeah, we'd have an unbound phosphate, right? So at the 5 prime end, we'd have an unbound phosphate. I should have said. Cool. But what do we know about DNA? That's what stranded DNA looks like. But DNA is, it's double helix is its shape, but to be a double helix, it has to have 2 strands. DNA is double-stranded. And that's noteworthy. Don't worry, we'll come back to that, because what will be across from this adenine? Guanine, good. And what will be across from this guanine? An adenine. And what will be across from this guanine? Is that true? Are A and G together? Adenine and guanine are both purines, pure as gold. Are they the ones that pair up?
Lisa Baltich, please return to class.
Because actually a purine always has to pair up with a pyrimidine. So adenine can't pair up with guanine. They're both purines. Adenine is designed to pair up with which pyrimidine? Thymine. What does guanine pair up with? So adenine pairs up with thymine. Guanine pairs up with cytosine. This guanine will pair up with cytosine, which means this thymine will pair up with adenine, and which means that this cytosine will pair up with guanine and guanine. Okay, but now this is actually key important stuff, and you wouldn't necessarily know this, but the second chain, don't draw yet. Everyone put your pencils down. Put them down. Is it going to go the same way? Is it going to look like that? Or is it going to go like, oh God, is it going to go like this?
Oh, okay, it's an upside down.
Yeah,
I had this pretty structured drawing in my old notes.
I love it.
With colors.
Love colors. I do love colors, big fan. And what we get is actually this structure here. And this is important because DNA is what we call an anti-parallel strand, which means that if on this side the 5 prime is at the top, on this side the 3 prime is at the top. Yeah, now you can draw it. All right, which means, oh, that was like the worst job ever drawing a bond from carbon to 1. That's not much better, but that's okay. We're going to call that good enough. Guys, why is drawing so hard? Other people seem to be so good at it, and I am not, and it makes me sad. That's the end of my story. And what is this down here? That's the 5' carbon, right? So, if one chain is running in the 5' to 3' direction, then the one that is bound to it is running in the 3' to 5'. They are anti-parallel. Parallel meaning they run beside each other, anti meaning they run in opposite directions. Does that make sense? If the 3' carbon is facing up on one side, the 5' carbon is facing up on the other.
It's like traffic.
Okay. What holds the two strands together? I've drawn them here as pink highlighter. But of course, it's not pink highlighter that holds two strands together. It's pink on my screen. What's it look like up there? Just kind of nondescript, I guess. Anyway, it's pink. What is that pink highlighter representing? Sorry? Did you say love? You guys don't know this, but what holds your DNA together is love. Love. Ooh, love. Has anyone seen The Princess Bride? It's a great movie. It's a perfect movie. You should all watch it. No, but do you actually think that? That's a terrible opinion to hold. I have less respect for you now than I had for you moments ago.
I'm fine with that.
No, you're not. You can say that all you want. We both know it's not true. You hold my opinion in high esteem, and as you should. So yeah, it's actually not love. I mean, it might be love. You tell me. I don't know. Is the fundamental force of the universe love? Maybe. But you know what holds things together? Jesus in Sunday school. What's the answer? Hydrophilic interactions in this case. In other words, good old-fashioned hydrogen bonding. All right? Hydrogen bonding, force of attraction, van der Waals forces, all those things if you learned about in chemistry. But long story short, these nitrogenous bases are polar, and so they're attracted to each other, and we call that hydrogen bonding. All right? Just dipole-dipole interactions. This is polar, that's polar, they're attracted to each other, which means these two chains end up being stuck together. But there's no bond between them. Does that make sense? There's no bond between them, merely a force of attraction, which I guess could be comparable to love. You know, you're not physically stuck to your partner. It's the bonds of love that holds you together. The DNA chains aren't physically held together. It's the bonds of, you know, dipole-dipole interaction. And really, in the end, what's love but a dipole-dipole interaction? You can put that on my tombstone. In the end, imagine that, like people just walking by. In the end, what's love but a dipole-dipole interaction, I think, really. Anyway. All right, so hydrogen bonding between nitrogenous bases holds the two chains together, two strands together. Oh God, double L, E, single L, right? That looks wrong, but I think it's right. Is it right? It looks wrong, right? Can we all agree? So if this is in the 3' to 5' direction, then the second strand will be in the How do you think we figured out that A and T bind to one another and G and C bind to one another?
We stole some DNA.
Okay. You have to steal it. Easy to get DNA. But seriously, like, are we able to look at DNA under a microscope and see the individual molecules? No, microscopes aren't that sensitive. So how do we know this? It's actually a great observation. So we know about complementary pairing. So we know that adenine always pairs up with thymine, and guanine always pairs up with cytosine. And in grade 11, we tell you that's true, and we make you practice a bit. And I probably on a test, I bet if I taught you, I probably gave you a line of code. Like I probably said, make the complementary strand, right? And you had to write the complementary strand underneath. And you just knew that A goes with T and G goes with C. How do we know that? Right? Who was the first smarty pants? Sam Brady, original smarty pants. Put that on your grave. Down the road from mine, which is in the end, what is love but a dipole-dipole interaction? All right, well, the original smarty pants in this case is a fella named Erwin Chargaff. I never know how to pronounce his last name. Or Chargaff. I don't know how you say that. I've only ever read it in a book. What do we think?
We'll just call him Erwin C.
Does he have something named after him too?
Yeah, a law or a rule. In his case, we call it a rule. I don't know why some are rules and some are laws, but his is a rule. But he researched DNA from different species in the 40s, the 1940s.
He was doing that while World War II was ending?
I mean, yes, I guess.
It's like he was fighting.
And what he found was that different samples of DNA had different proportions of A, G, C, and T, which makes sense, right? But this is actually before we knew the structure of DNA, but we knew what it was made of. We knew that it had different proportions of A, G, C, and T. The proportions of sugar and phosphate were always the same, right? Now, we happen to know that this gives a sense that what part of the DNA molecule must be the code. What he finds is that if you take a carrot, you take a person, you take a monkey, you take a pea plant, they always have the same amount of phosphate and the same amount of sugar, so there's no information to be had there, right? But this stuff comes in different proportions. That gives us an indication that that's the part of the DNA that's acting as the code. And so Chargaff knows this. Other people figure that out too, but he discovered a second thing. But, and they say you're not supposed to start a sentence with a conjunction, but here we are, and I just did it. And you can't stop me. But if you do it too often, it gets distracting. And that doesn't stop me from doing it. So let's get to work. Bless you. But he found that the proportion of A and T not plus, that implies added together, that the proportion of A were always the same. As were G and C. This is called Chargaff's rule. Again, I'm not sure how we pronounce that. And this gives us the sense that they must be paired, right? Who actually gives us the structure of DNA? So Chargaff gets us part of the way there. Chargaff gets us to the idea that A, G, C, and T must be the Cody bits of DNA because it's their proportions that are varying. And he gets us the important bit that these must kind of pair off in some way. Does that make sense? That's the logical conclusion from this. If the proportions are always changing, but A and T are stuck together and G and C are stuck together in proportions, then they must be stuck together in the molecule, right? That if you have one, you must have the other one. Who are the scientists who actually give us the structure itself? Famous. One day, no doubt, but not today. Watson and Crick. Watson and Crick. So by the time Watson and Crick discovered the structure of DNA,
Oh. Yeah, he was more than just a journalist. Author. That's two people?
yeah. It'd be a weird name if you just named your kid like, this is my kid, Jerry and Tom. Like, oh, you have two? No, it's one kid.
It's like Mary Ann. Two names, but it's one name.
If it's not Mary and Ann, which would be a whole different thing, right? You guys should just all do that. Give your kid names where it's two names with an and between them. It's my son, Larry and Bill. Oh, your son's a? No, just one. That's Larry and Bill. Try to pay attention.
To be a servant? With my 7 kids. It's too much. I'm too sensitive. I think that's weird.
Okay, so by the time Watson and Crick discover the structure, we know that A always pairs up with T and G always pairs up with C, right? So thus, the two strands are complementary. And if you know the code of one strand, then you must know the code of the other. Which also means that you can't really write code on both of them, right? We usually refer to one strand of a section of DNA that has a gene on it as the coding strand, and then we refer to its opposite as the complementary strand. Interestingly, it's not always one or the other. Does that make sense? It's not like one side of the DNA is always the side that codes and the other side is always the side that's complementary. On the physical molecule itself, sometimes the gene is held on one side, sometimes the gene is held on the other, pretty much at random, pretty much in equal proportion, which is interesting. The shape of DNA is famous. The double helix. This is because if you look at the structure of a DNA molecule, you have, God, where are we? You have this section here that is hydrophilic enough, all right? The nitrogenous bases are fairly hydrophilic, but what is on the outside structure of all these? Phosphate groups. And phosphate groups are incredibly hydrophilic. So they have a much greater affinity for water than does anything in the middle. Does that make sense? Which means that if you surround this with water, or any other polar solvent for that matter, the phosphates are going to orient themselves such that they maximize contact with water, which means that the bases are going to orient themselves in such a way that they minimize the contact with water. And that isn't if you just keep the thing straight like a ladder. Does that make sense? If you keep it straight like a ladder, the phosphates are in contact with water at exactly the same proportion as you can imagine like the rungs of the ladder in the middle. Does that make sense? So the rails are no more in contact with water than this. But what if you take that and you twist it? Now all of a sudden, the whole outside of the molecule is the phosphates. Does that make sense? And the inside of the molecule now becomes the bases and the sugars, like the interior side of the sugars, like the 1' and 2' side. Does that make sense? So picture a ladder.
No, I get it. I
Yeah.
just don't understand that if you like give it its helix form,
Yeah.
like it's still technically like just as much touching of the water.
But relatively speaking, it's actually not, which is to say, right now, a water molecule is just as likely, if a water molecule flies at this thing, it's just as likely to hit here as it is there. Does that make sense? Which means if you imagine a water molecule shooting at this thing, the phosphates actually make up a relatively low percentage of what they'd come into contact with. Yep. But if this thing twists in on itself, now the phosphates make up an outside of the molecule and the bases make up an inside of the molecule. Laying flat, it doesn't have an inside and an outside. It's kind of linear. But if you twist it, right, no different than if I take this piece of paper, right now, right, there's no inside or outside to it, right? It's just all equally in contact. If I go like this, now it has an inside and an outside, right? Sorry?
That's not like the shape it's making.
Well, that's because this is just happens to be something I had on my desk, right? But now if you picture a ladder, again, it doesn't have an inside and an outside really. It has a front and a back and sides, right? But that's equally accessible from every direction. But if you twist it, now that's not true, especially if the sides are fairly thick. All of a sudden now, picture a spiral staircase, and if you throw something at it, you're much more likely to hit the rails of it than you are to hit the steps of it, because it's spiraling like this, right? So if you spiral it, the edges end up being basically the only thing in contact with the outside world, and the inside of it. And if you picture a spiral staircase, there's like one place you have that there isn't railing, so you can go up and on it. But then you really can't get off the staircase. You can't get from the inside to the outside without like hopping over a railing, which is a bad idea because you're on a staircase. But does that make sense? You've effectively created something that now has an inside and an outside, which is different. This is, by the way, why we use a spiral staircase, not a ladder, to get up to things, because it's much safer, right? Because it has an inside and an outside, as opposed to just having steps that you can easily fall off. Does that kind of make sense? Can I see you climbing a ladder motion again? I like that. I know, but yours is much cuter. That was good. I liked that. So the double helix forms naturally by virtue of Jesus at Sunday School? Hydrophobic interactions. So in this case, hydrophilic interactions, right? But it's all just hydrogen bonding. So hydrogen bonding between PO4 groups and water induces DNA to twist into its characteristic double helix shape. That is like one sickly looking strand of DNA, but it's going to have to do. Say it again.
No, it's fine.
No, no, no.
I just don't understand. Like, no matter how you twist it, it's still surrounded by fluid. So how does it like shield it or create like a shape?
No,
I don't
like
really.
no different than like if you went in a swimming pool. Not that it's going to keep you dry, but water is going to have less contact with, say your chest and stomach if you went like this than if you go like this, right? Like, so in other words, yeah, you're still in contact with fluid surrounding it, but now keep in mind that the difference here is that like a water molecule is sizable compared to this. So when this twists in, the inside of this now isn't in contact with water. You know what I mean? Because this isn't like this in like you can't think of water as like a liquid that moves through it here. Like a water molecule would be like, you know, a sizable thing like this here. And so when this twists into a double helix, the result is that the outer chain of sugars is much bigger than the water molecule and is now twisted on itself. Water isn't getting to the inside and contacting the bases. So like at this point, you do have to think of it as being at a small scale, not kind of picturing it at our scale. Now, water can still have some interaction with the bases, but this will really shield it. This will really perform interference. And more importantly, it's not about shielding the bases. What it is about is that the phosphate groups are highly attracted to water, and the reason it twists into this shape is that it maximizes its contact with water, which is energetically favorable. Basically, phosphate is always trying to get as close to as many water molecules as possible, and it's able to get closer to more water molecules by twisting it into this shape than by laying flat, and so it's energetically favorable to twist into this shape. And so it does. Nope, I changed colors. And again, note that they're anti-parallel. What's anti-parallel mean again? Sorry? They go opposite ways. In other words, we draw this as a line for convenience sake, but it's not a line. It's actually a big chain of very bulky sugar molecules, and they are arranged upside down relative to one another. So in this case, if the 3' OH side of this sugar is facing up, then the 5' PO4 group of this sugar is facing up. They are in opposite orientation to one another. How's DNA built? Sorry? This unit is called molecular genetics. It's a good title, eh? I can't stand the auto underline thing. Will make lines that are just ever so slightly not straight. When do you ever want that feature? When do I ever want a line that is just ever so slightly not straight under my... Do you know what I mean? Like that one's good. Anyway, okay. When DNA is polymerized, by the enzyme, does anyone know the name of the enzyme? Yes. Yeah, helicase takes part. When DNA is polymerized by the enzyme DNA polymerase, clever name, we can all agree. New nucleotides are always added not always sadded, but rather always added to the OH on the 3' side of the previous nucleotide. The other way we can say this is just that DNA is built in the 5' to 3' direction. And if you think about the fact that we have two chains, this is going to be important when we build DNA, because one of the chains builds really easy, another one of the chains is going to build less easily, right? But DNA is always built in the 5' to 3' direction. That's how the enzyme that we have to build DNA works. So this is important and challenging. When making double-stranded DNA. It's actually really noteworthy because what the cell could not do is sit and build the strands together at the same time. Does that make sense? Because the builders would want to go in opposite directions, which means that one strand has to already be present for the second one to get made against it. Does that make sense? The strands get built one at a time. Or, as we'll see, it's always a function of the fact that we're always working from DNA that already exists. We're always building off a template as opposed to just building DNA fresh. We're going to leave it there today. I think that's a good amount of information. No homework for today. I'll have some readings for you after our next lesson, but we'll take a break. I don't know about you, but every once in a while, nice to have a break. Oh my goodness.